Hello, and thanks for joining us today for the latest Technology Networks webinar, Generation and Functional Characterization of Human-Induced Pluripotent Stem Cell-Derived Cortical and Sensory Neurons. I'm Karen, Science Writer for Technology Networks, and I'm here to moderate the event. I'm very pleased to have Dr. Giovanna De Filippi from Axion Biosystems and Dr. Stephen Broadbent from Axel Bioscience joining us today as your presenters. A very warm welcome to you both. Giovanna is the Business Development Manager, EMEA, of Axion Biosystems, supporting and implementing growth opportunities for their complete line of instrumentation. She holds a PhD in Physiological Sciences from the University of Milan, Italy, with a focus on neuroscience research. In her career, she has gained 10 years' experience in neuroscience research and preclinical drug discovery within the pharmaceutical industry, and 10 years' experience in scientific and commercial roles in the instrumentation industry, serving the life science and material sciences markets. She has a wide understanding of customer needs in both scientific and business settings, and a passion for bringing novel techniques and instrumentations to scientists. Stephen has nearly 20 years' experience in electrophysiology and a background in pharmacology, neuroscience, epithelial and cardiotoxicity, including 5 years working with multi-electrode array (MEA) systems. He's been with Axel for 2 years, during which time he's worked extensively with Axel's in-house hiPSC-Derived neuronal and cardiac cells. This has included differentiating and culturing them and validating their function in monoculture and co-culture in 2D and 3D assays using MEA systems. Initially, he was an electrophysiologist on the MesoBrain project, an EU initiative to develop and validate a 3D culture of 4 of Axel's cortical cell types: excitatory cortical neurons, inhibitory interneurons, astrocytes, and microglia, suitable for MEA and calcium imaging assays. For the last year, he's been responsible for a number of bespoke MEA projects, assessing the pharmacology and phenotype of different Axel iPSC-derived cell assays for external collaborators, as well as providing technical support to Axel's commerce team. Following the webinar, we will have a Q&A session and welcome any questions that you may have. You can submit a question for the Q&A session at any time during the presentation. To ask a question, please click the chat icon in the bottom right corner, enter your question into the box on the right-hand side of your screen and click Submit. We will answer as many as possible during the time available. For any questions we don't have time to get to, we will ensure you are contacted offline with an answer. Please remember you can ask questions at any time, even if you're watching this webinar on demand. So without further ado, I will now hand over to Giovanna and Stephen.
Thank you, Karen, for the kind introduction. Good afternoon, good morning, everyone, and thank you for joining this webinar today. My name is Giovanna De Filippi, Business Development Manager for Europe at Axion Biosystems. Axion Biosystems is a global leader manufacturer of microelectrode-based technologies for biomedical research. The focus at Axion continues to be on developing a platform of fully integrated and scalable microelectrode array products which are easy to use, reliable, predictive, and available in multi-well plate format. We will discuss here the key features of the Axion MEA platform and will focus on measurement of neuronal activity, how to characterize neuronal networks, and establish model of a disease in a dish. A microelectrode array is a grid of extracellular electrodes embedded in the substrate of a tissue culture dish or plate. Neurons or cardiomyocytes are cultured on top of the electrodes. When they start firing action potentials, these are detected extracellularly by the electrode array. Therefore, MEA allows for electrophysiological recording of electroactive cells in a non-invasive and label-free manner. The 2D distribution of electrodes in the array allows to detect and characterize the formation of neuronal network in the culture, making this technique very powerful, as standard higher content electrophysiological techniques can only record from one neuron at a time, losing all information on network activity within the preparation. The Maestro Pro and Edge are the flagship product of MEA recordings. They differ for the total number of electrodes, which affect what format of MEA plates can be used. Maestro Pro supports the whole range of MEA plates, while Maestro Edge can only read from 6 and 24 well plates. Otherwise, all hardware and software features are the same. Both are a benchtop size instrument with a built-in incubator. The assay starts with the push of a button to dock the MEA plate in the Maestro. This triggers the adjustment of the temperature and CO2 levels and the automatic recognizing and logging of the MEA plate type and number via an integrated barcode scanner. All 768 or 384 electrodes can record the data as well as be used for electrical stimulation. In addition, thanks to our proprietary electronic chip design, the same electrodes can be used to measure intracellular potential-like signal from cardiomyocyte, impedance as a surrogate of cardiomyocyte contractility, or impedance from non-electroactive cells to study cell proliferation or intracellular signaling, for example, for cancer-related assays. Both instruments work with Axis Navigator software for MEA recordings, together with a series of standalone tools for advanced data analysis and automated plotting of results, and they also support the Axis Z software for impedance recording. Both instruments can be used together with our Lumos range of products for pairing MEA recordings with optogenetic stimulation. The neural module of Axis Navigator software has been designed to provide a straightforward and simplified approach to the setup, execution, and analysis of neural MEA experiments. Pre-configured settings for data acquisition and analysis are available. Axis automatically shows the type and format of the plate in use and provides an interactive plate diagram that allows to create color-coded plate maps with information such as cell type or pharmacological treatment for each well. As the experiment runs, Axis Navigator allows multiple views of real-time activity on any of the well in the plate. In the continuous waveform plot window, you can monitor the continuous plot of the raw voltage from each electrode in well C1. Each rectangle here represents one electrode in the well. In the spike plot window, you can visualize the detected spike waveform on each electrode in the well, and at the bottom, you can follow in real time the well-wide raster plot where each line represents one electrode. The white marks are individual spike, the red marks indicate detected single electron bursting behavior, and the teal rectangles show detected network bursts within the well. In the activity map window, the whole plate is represented and each square shows a well. The activity, whether spike frequency or amplitude, is color-coded with warm color indicating a high value of either parameter. This provides an immediate visualization of the activity across the entire plate, allowing correlation to either the culturing conditions or the pharmacological treatment applied. The workflow to set up and run MEA measurement is very simple. Plate the neuron on an Axion MEA plate of desired type and format. Load the plate into the Maestro and allow the environmental chamber to automatically equilibrate. Use the neural module of the Axis Navigator to select the recording configuration and start data acquisition. At the end of the recording, you can position the plate back in the incubator and repeat the measurement daily to follow the network development and maturation. The Maestro MEA system detects key parameters of neuronal function to provide information about activity, synchrony, and oscillation. Activity of the neurons is quantified by measuring the frequency of action potential detected. Single electrode analysis provides information on individual neuron firing and bursting. Synchrony reflects the strength of synaptic connection established between the neurons in the cultures and how likely they are to generate action potential simultaneously on a millisecond timescale. Oscillation is a measure of how the spikes from all of the neurons in a well are organized in time and whether their firing is coordinated across the neurons in the network to provide alternating periods of high and low activity. Multiple electrode analysis provides information and endpoints to quantify network bursting and connectivity. Axis Navigator neural modules provide about 30 metrics or endpoints to characterize the behavior of a neuronal culture firing activity. They allow a detailed characterization and classifications of either a neuronal culture model or of effect of drugs or neurotoxic agents. In addition to Axis Navigator, the Neural Metrics standalone tool provides more in-depth analysis of aspects of the neural activity analysis, allowing, for example, finer tune controlled over burst, single electrode burst, and network burst analysis parameters, analysis of synchrony, and analysis of electrically or light evoke responses. In addition, the neural metric tool now supports unit-based analysis of spike waveforms that have been sorted via Plexon Offline Sorter, a third-party spike sorting tool. Data endpoints from analysis with the neural metric tool can be exported for plotting. The Axis metric plotting tool allows automated plotting of endpoint statistics from Axis Navigator and neural metric tool, and allows easy comparison across different data files or different time points. The plots and the related value can easily be exported. MEA technology is ideal to combine with primary or stem cell-derived 2D and 3D models. In the neuroscience field of research, the Axion MEA technology is adopted worldwide by scientists in diverse research environments, mainly for 4 different areas of application: characterization and quality control of stem cell-derived neurons, functional disease modeling of psychiatric and neurodegenerative disorders, phenotypic screening for drug discovery, and neurotoxicity screening, whether for potential epileptogenic effects of new chemical entities or for potential neurotoxic effect of everyday compounds. MEA data generated with Maestro appear in more than 250 peer-reviewed publications, and the number keeps growing. You can find most of them in the resource page of our website. Optimization and characterization of hiPSC-derived neurons are an essential first step when establishing robust and reproducible cellular models in a plate. Neurons take approximately 7 to 10 days before starting showing some spontaneous action potential after they have been plated. On the left here is a typical well-wide raster plot from MEA recordings of 2-week-old cortical neurons. Each line represents a different electrode in the well, and it is apparent that not all electrodes detect activity, and that the number of spikes detected is highly variable between electrodes. However, at 4 weeks in cultures, the neurons have reached a more mature stage of development and have formed stable synaptic connections. These reflect in a higher number of electrodes showing spontaneous activity, a higher number of action potential being fired, and network burst activity being detected, also indicated by the pink rectangles here. With Maestro MEA, it's possible to follow the maturation of the neuronal culture and the changing phenotype resulting from the establishment of stable synaptic connections. Optimization of cell density, culture protocol, media type conditions is made faster and more efficient thanks to the higher throughput allowed by the multi-well plates and the automated data analysis. As an example of how the Maestro is being used for functional disease modeling, here researchers have compared hiPSC-derived dopaminergic neurons from monozygotic twins carrying a gene increasing the susceptibility to sporadic Parkinson's disease, but with only one twin showing the clinical manifestation of the disease. At 30 days in culture, the spontaneous activity in dopamine neurons from the affected twin is significantly lower when looking at the activity map. By 52 days in culture, the same activity map showed increased activity for the neurons obtained from the affected twin. To further qualify this observation, the raw data traces and the wide well raster plot show that the overall activity is indeed lower in the neurons from the affected twin at 32 days in vitro, and this effect can be quantified by measuring endpoints such as mean firing rate or the number of active electrodes. In addition, by day 52, the same culture showed increased activity of dopaminergic neurons from the affected twin. However, this activity was disorganized compared to the mature network bursting phenotype observed in neurons from the unaffected twin, highlighting an absence or perhaps a delay to times beyond the course of these experiments of synchronous activity in the affected twin. Here I show one of various examples of experimental work published utilizing MEA recordings of rodent dorsal root ganglion neurons to model pain diseases. The group is researching rare mutations in the NaV1.7 sodium channel that are continuing to be identified in individuals suffering with inherited erythromelalgia, a condition for which there is no treatment available. In the case studied here, the patients were treated with carbamazepine, a sodium channel blocker, which showed to release the symptom in erythromelalgia patients carrying a different mutation of the same NaV1.7 channel. And indeed, carbamazepine reduced the pain symptoms in these patients also. So DRG neurons and sympathetic ganglion neurons express NaV1.7 channels. So here, rodent DRG neurons were used to induce the expression of the mutation, and their activation by heat was assessed with MEA recordings. Increasing the temperature from normal skin temperature, 33 degrees, to core body temperature, 37, to a non-noxious warmth, 40 degrees, increases the number of DRGs being activated, as well as the firing frequency of the neurons. However, in the presence of 30 micromolar carbamazepine, a clinically relevant concentration of the drug, both the number of active electrodes and the firing frequency are markedly reduced at all three temperatures, and the white arrowhead here indicates silent DRGs after carbamazepine treatment. So the DRG neuron preparation offers an experimental model to screen for new drugs for pain-related diseases. In summary, we learned that the Maestro Pro and Edge MEA platform provides label-free functional assays of neuronal preparation, comprehensive automated analysis of neural network activity, a user-friendly and powerful tool to reproduce human biology in vitro, characterize neural activity, and a tool that can be effectively utilized to generate disease-in-a-dish models of neurodegenerative, pain, or psychiatric disorders. With this, I would like to thank you for listening and look forward to your questions after the next presentation. Thank you.
Hello, my name is Dr. Stephen Broadbent. I am the Senior Scientific Support Manager and Resident Electrophysiologist at Axel Bioscience. Today's talk, I'll be discussing how Axel's iPSC-derived sensory neurons can be used to enhance your research. I'll be talking about how to use the cells and sharing some functional data we've obtained using the Axion Maestro Pro with our cells. I'd like to start by just giving a brief overview of today's presentation. I'll begin with a very quick overview of Axel as a company. I'll discuss the launch of Axel's new MEA screening service. I'll share some validation data of Axel's iPSC-derived sensory neurons with our proprietary maximizer supplement. But the bulk of today's talk will be looking at MEA recordings from Axel's cells using the Axion Maestro Pro. And hopefully this will give some insight into how Axel's iPSC-derived sensory neurons can assist your research. And as such, I'll be giving lots of examples to do with compound screening. I would like to begin by giving a quick description of Axel Bioscience as a company. We were founded in 2013 by Dr. Yichen Shi. We are located in Cambridge in the UK, but we have a logistics facility in Boston, Massachusetts in the US, and we are currently expanding our footprint within the US. We have a very large product range of iPSC-derived cells, along with all their supporting reagents and supplements, and these all come with full technical support and protocols for their use. We see ourselves as a one-stop shop. We can take your cells from primary, fibroblasts and PBMCs, we can reprogram them, gene edit them, differentiate them into a range of end cell types, we can bank these, and increasingly now we can perform end-use assays for you. And our business model, we see ourselves helping you at any stage in this. We can take your cells at any point, to any point, whatever you require. It's an exciting time at Axel with the recent launch of our new MEA screening service. As I mentioned, we have an extensive catalogue of iPSC-derived cells and a wealth of experience of culturing these both individually and in co-culture. When we combine this with over 20 years worth of experience of electrophysiology and the recent purchase of the market-leading Axion Maestro MEA platform, these factors all combine to put Axel in a perfect position to offer a unique service, which we're very proud of. I mentioned our very large range of products, and I'll give you a quick overview of the sort of cells that we work with and can provide. Our company was founded on the use of iPSC-derived neuronal cells, and this remains a significant part of our portfolio. We have cell types from both peripheral and central nervous systems. We have sensory neurons, which with our maximizer we can take through to maturity within as little as 3 weeks. And we have a wide range of cells from the central nervous system, including cortical neurons, both healthy and disease lines, astrocytes, microglia, striatal neurons, and motor neurons. And we have a healthy pipeline with a whole range of additional neuronal cells coming through in the near future. As well as growing these cells individually, we have a wealth of experience growing these in co-culture. In addition to our core neuroscience business, Axel's also got a strong interest in the cardiovascular field. We supply hiPSC-derived ventricular and atrial cardiomyocytes and endothelial colony-forming cells. The atrials and ventriculars come from the same donor, which makes them ideal for doing any comparative studies, and the endothelial colony-forming cells are great for modelling angiogenesis or any sort of form of co-culture with our other cardiomyocytes. And these cells are ideal for a range of uses. However, that's not the focus of today's talk, but please keep your eyes peeled for the accompanying talk on our cardiomyocyte offering. As I'm sure you're all aware, sensory neurons carry sensory information to the CNS. Here we have a cartoon of painful stimulus being transmitted through the sensory neuron to the spinal cord and triggering a muscle reflex. Sensory neurons are also involved in pain, thermoception, inflammation, itch, pain pathologies, and neuropathies. So they are and remain a significant area of interest. Axel Bioscience's hiPSC-derived sensory neuron progenitors were launched in 2018, and while they're fine, one of their issues was it used to take up to 70 days to reach functional maturity and show correct response to things like capsaicin. So last year we launched the Sensory Neuron Maturation Maximizer. This is a supplement for our standard media and reduces that maturation time from 70 days down to 20. All it essentially does is mimic the extracellular milieu the sensory neurons would experience during development, and we replicate that. And as you can see here, you get standard responses to capsaicin at a much earlier time point with that. In addition to the functional data, things like ICC show marked increase in the key sensory neuron markers, such as VR1, the TRPV1 channel. Our maximizer also produces this lovely tangle of processes marked up by TUDGE1, and you can also see that the NAVs, the sodium channels involved in pain, are very well expressed as well, even at day 24 compared to day 70 previously. Along with ICC, Axel has had RNA-seq data produced for all our major cell lines, including our sensory neurons. Here you can see that our mature sensory neurons, the neurons which have been treated with our maximizer, very closely resemble the RNA-seq you get with the human DRG, and we're very happy with what close a match we get with our cells. If there's any sort of targets you're interested in which you don't see there, please just ask us and we will take them out and pass the information on to you, no problem. Now, pretty pictures are fine, but I'm an electrophysiologist and it's the functional data that I'm really interested in. I'm sure you're all very familiar with this. It's a cartoon showing the neuronal action potential, showing how sodium channels and potassium channels open and close, driving that standard sort of action potential you would obtain via something like patch clamp. Now, patch clamp's great, but it is technically challenging and its throughput is very low, which is why Axel Bioscience have focused on the MEA platform so much. Now, MEA systems, if you're not familiar with them, don't measure action potentials, they measure field action potentials. The field action potential is like the derivative of the action potential. It's essentially the slope. So each part of this waveform corresponds to parts of the action potential. You have the sodium influx here, followed by potassium efflux there. Although the neuronal field action potential does look a lot different from the neuronal action potential, the distinct parts of the waveform do correspond to real things within the action potential, meaning examining the field action potential can reveal information about the underlying pharmacology of the cells. Similarly, with the MEA system, you can look at spike rate, burst firing, and network level activities across a well, things you can't do with standard patch clamp. So as such, I'm going to show you some real data we've obtained using our cells on the Axion Maestro Pro. So we'll just press play. These are sensory neurons after 20 days culture. And as you can see, they're very active. Every electrode in this well is firing very quickly. If you look at the heat map, which shows general activity, you see most wells, most electrodes are firing quickly. The brighter the lights, the more activity there is, and they are extremely active. We're very happy to see this. You can then zoom into an individual well, and as you can see, every electrode in the well is firing very quickly. And the third panel will zoom into individual spikes, and you can see it constantly updating field action potentials coming from each electrode. The red squares show bursts of at least 5 spikes in quick succession from different electrodes. So as you can see, very active cells. So that was day 21. Now we're going to look at day 28. So again, another week's culture within our maximizer supplement. Press play again, and you see the activity seems to have dropped off, but rather than individual activities, you're getting these flashes which correspond to these bursts across multiple electrodes at the same time, spontaneous burst firing. The entire network is activating as one, which is what these sort of red columns are. And as you can see, every 20 seconds or so, you'll get a big burst of activity across most of the electrodes in a well. Now that was showing the data, just baseline data at day 28. This is now what happens if you add some capsaicin. So again, you're getting the flashing. If you wait a second, it'll happen again, I'm sure. There we go, lovely. If you zoom in, you can sort of see that on the electrode traces. And most interestingly, if you go to the raster plot, you can now see these sort of solid columns of red much more frequently than you were getting just with the baseline. The capsaicin is inducing this activity across the entire network, which is very impressive. So as we see, the Axion Maestro Pro is an ideal platform for examining the electrical activity of our cells. And from that, we can extract a large amount of data. We can pick out traces and spike rates, well activity. We can pick out example waveforms, raster plots, frequency distribution histograms. And if you drill into the numbers, there's a whole range of other parameters you can look at. And Axel have developed 76 parameters we can examine by drilling into these numbers, covering a wide range of different characteristics. Now, if you look at the Bradley et al. Paper of 2018, they showed that a lot of these characteristics do correspond to neurotoxicity, which is great. So it means these cells are ideal for looking at pharmacology, pathologies, and neurotoxicity. I can assure you my electrophysiology skills are much better than my PowerPoint skills. Where the Axion Maestro and Axol's sensory neurons with our maximizer come to their own is in compound screening. You culture our cells, after 3 weeks you get nicely active cells which respond well to compounds of interest. Here we have capsaicin. After 20 days you're getting a big increase in spike activity, as you would expect. Now you can plot this using spike frequencies, showing how the activity builds with concentration. Or you can zoom in and look at the waveform. Each aspect of the waveform corresponds to a different part of the neuronal action potential. You have that initial depolarization where sodium enters, a real polarization as potassium leaves, and then the hyperpolarization and recovery phase. From this, you can measure a whole load of different parameters: field potential duration, first and second peak amplitudes, rise times and slopes, areas under the curve. Then it's endless. Now, as it happens, if you look at capsaicin, you don't really see much change to the waveform. This tells us that there's very little off-target effect, and all the capsaicin is doing is just modulating the underlying activity of the cells. And you can demonstrate this through looking at like a radar plot. Here we have 6 key parameters to do with waveforms, and as you see, the difference between the control and the capsaicin-treated cells is minimal. There is no real difference in the waveform parameters. Now we can also look at raster plots. Raster plots pick out individual spikes, they will pick out individual bursts, and if there's activity across the well, it will pick out synchronized burst firing. And whereas looking at capsaicin's effect on the waveform produces very little difference, looking at its effect on these key burst parameters, you can see there's a very marked difference between the cells which are treated with vehicle control and the cells which have been treated with capsaicin. And we can repeat this for a range of classic compounds. So you can see here, spike increase with menthol, mustard. Now mustard is interesting. There is a small increase, but it's very marginal. So it doesn't really look like it's doing much in terms of affecting spike rate. However, if you look at the raster plots, you can see that 100 micromolar mustard oil is actually producing much more in the way of synchronized burst firing across the well, even though the underlying activity isn't actually changed that much. And we've looked at a number of different compounds using our cells. ATP is very interesting. This is a 60-second trace, and as you can see, ATP produces very short, intense bursts of activity, which stops very quickly. We've looked at glycine. You can see the sort of slow onset of glycine activity on our cells. And here we have histamine, standard sort of itch and inflammation mediator. And as you can see, on our sensory neurons, you're getting a massive increase in activity when exposed to histamine. In addition to looking at the pharmacology of the sensory neurons, our cells are also great for looking at how they respond to noxious stimuli. Here we have in day 23 our cells responding to a cooling ramp. You can see as the temperature drops, the cells stop firing. Warm it up again, they come back. Similarly, if you increase the temperature up to 45 degrees, you can see a marked increase in activity, which is brilliant. It shows the cells are responding in the expected way to a noxious stimuli. I hope you enjoyed that brief taster of what our sensory neurons can do. That is only the tip of a very large iceberg for us. In future, we're going to continue adding extra compounds, and we are slowly building up a panel because we see an opportunity here for performing neurotoxicity studies. Our other talk discusses cardiotox, but also we see that our cells could be ideal for assessing potential neurotoxicity. And it's not just our sensories. We have a range of other neuronal cell lines. We have corticals and dopaminergics and striatals. They're all perfectly applicable on the MEA platform. We also have a large number of disease models. And we can always add more, either deriving them from patients or gene editing them ourselves. And finally, we have got a wealth of experience doing co-culture. I mentioned our cortical neurons. These can be co-cultured with our astrocytes or microglia, our GABAergic interneurons to produce a much more physiologically relevant model. So in summary, the combination of Axon's Maestro Pro and Axel's unrivaled catalogue of neurons and supplements such as the Maximizer means we have an exciting opportunity here to assist you in your neuroscience research. And our newly launched MEA screening service allows a unique synergy of a great amount of expertise to produce a service offering we are very proud of. Thank you for your attention, and if there's nothing else, we'll move on to some questions.
Thank you both very much. I'm sure you'll all agree there have been some excellent points raised during the presentation. So we'll now begin the Q&A session. To question to our speakers, please enter your question into the box on the right-hand side of your screen and click Submit. We will answer as many questions as possible during the time available. So we've had a few questions come in already, so we're going to dive straight into the first one. This one I believe is directed towards Stephen. Do you perform MEA assays using other cell types?
Yes, so MEA is suitable for all of Axel's cardiac and neuronal cell types. So we have atrial and ventricular cardiomyocytes, we have sensory neurons, cortical neurons, striatals, dopaminergics, motor neurons, interneurons. These can be both wild type or disease lines, either from patient or genetically engineered. We have co-culture models using astrocytes and microglia. And yes, so any of the sort of standard like electrical cells, Axel cell would be suitable for the MEA assay.
Brilliant, thank you very much. And how much does Axel's MEA service cost?
So our pricing tend to be bespoke, so we would discuss this with the customer first. But as a baseline guideline for a basic 48 well plate, cardiomyocyte plates starts at £2,700 each, and the turnaround there is about 3 weeks. Cortical neuron plates, just monoculture, is £5,725 each. They take about 5 to 6 weeks. The sensory neuron plate is 7,250 each. That's with the maximizer, and that's ready in 3 weeks. Obviously, any additional analysis or calcium imaging or co-culture or longer culture would add to the price, but that would always be discussed with the customer first. Thank you.
Brilliant, thank you very much. For our next question, for how long can iPSC-derived neurons be monitored in culture with Maestro MEA?
So they can be monitored for as long as your culture remains viable. We have customers that follow the network properties for up to 70 to 120 days. So really there is no limit apart from the healthy status of the culture itself.
Brilliant, thank you very much. And for the next question, what plate format is most commonly used for hiPSC neurons?
For neurons, the most popular plate format seems to be either the 24 or the 48 well plate. Of course, with the 48 well plate, you will be able to do more replicates within one experiment, or you will be able to test more conditions and compare more conditions within the same plate. For a Maestro Edge, the maximum format available is the 24. So a lot of studies are also run in a 24 well plate.
Excellent, thank you very much. What is the time course of NAV 1.7 and NAV 1.8 expression? And is this accelerated by the maximizer media?
Yes, thank you. So yes, the NAV 1.7 is 1.8 with the maximizer. The ICC images I showed earlier were from week 3. That's with the maximizer. Without the maximizer, you would have to take the cells up to at least week 6 to see comparable levels of expression. Thank you.
Excellent, thank you. So this brings us to our last question that we have time for now. Was this measured with the Maestro?
Yes, so the data I showed here was a screen grab of the Maestro in action on the axal cells. So all that data from that initial part of the presentation was recorded from the Maestro. Thank you.
Excellent, thank you. Once again, a big thank you to you both. That's all we have time for today. Just to remind everybody that any questions we didn't have time to get to, you will be contacted offline with an answer as soon as possible. You can ask questions even if you're watching this webinar on demand. It will now be made available on demand, so if you've got friends or colleagues that you think may be interested, then please feel free to share the link. Attendance is available to download from the Handouts tab on the right-hand side of the webinar platform. And if you're looking for supporting material from this webinar, please also navigate to the Handouts tab. Thank you again for your time.